All right, guys, this is the second lecture covering topics 2.1 and 2.2. So the notes packet that you have been working through with organelles, this picks right back up in the next set of organelles. So the next organelle we're going to talk about is the mitochondria. Now, I'm thinking that you have probably heard about the mitochondria in some capacity in a previous year. So not only do we need to know its structure, which is fairly complex in terms of an organelle's structure, but then also what it does. So structure first, because structure determines function. Now, the mitochondria is an organelle that actually has two membranes. So it's called a double membrane bound organelle. There is an inner membrane, and then there is an outer membrane covering all of this. And so in the picture, you can see that the inner membrane actually creates this little pocket that's going to hold some fluid, and it's folded up inside to really cram more of it into the mitochondrial space. The zigzaggy folded pattern of this inner membrane is called the cristae. And again, this allows a lot of inner membrane to fit into a small amount of space. So essentially, if we highly fold in these cristae patterns, the inner membrane, we can get a lot of it in the overall mitochondrial structure. Now, that should give us a clue. If we want a lot of that inner membrane, there must be something pretty important, pretty critical happening related to that membrane, because we want a lot of it. And so stay tuned on that. But essentially, this bullet right here says that the highly folded cristae pattern is there to increase the surface area of that inner membrane, again, because something important is happening here. And then you have something called the matrix, the mitochondrial matrix, which is this fluid, very similar to cytoplasm in the overall cell. You have this inner fluid in this internal space. Now, what does the mitochondria do? Most of you have learned it is the powerhouse of the cell. That is not wrong. However, we are in AP Bio. We are going to broaden that. What does mitochondria do? It makes ATP. So ATP is going to be our critical, important cellular energy molecule, and most ATP is being created right here inside of this mitochondria. Something that we will find later that is oftentimes not understood or is a misconception is that plant cells also have mitochondria. In addition to animal cells, plants will have mitochondria and this next organelle that we are going to see. So the mitochondria and then the chloroplast here next, these are two really complex organelles that seem to be very similar to each other. They're ultimately associated with energy production. They are these complex organelles that have double membrane structures and some stuff inside. And so let's dig into a chloroplast. Structurally, again, a double membrane bound organelle. Good news, those membranes have very logical names, the inner membrane and the outer membrane. I believe you have a picture of chloroplast. It might not look exactly like this chloroplast, but it's very similar. And these are components that you need to make sure that you have labeled on your diagram. So inner and outer membrane for the chloroplast. Then there are these little discs, and the discs stack up into these towers that we see inside the innermost portion of the chloroplast. So one disc is called a thylakoid disc, and what's so important here, these discs contain the pigment that is essential in capturing sunlight and starting this energy conversion process in a chloroplast. So we know that chloroplasts are in photosynthetic organisms, and that means that those organisms can use the sunlight to create their own energy source. And so the actual conversion of light into chemical stored energy, sugar, is happening because of this pigment. And so the pigment is inside the thylakoid discs, which incidentally have their own membrane surrounding them. And so we find the pigment there in the membranes of the thylakoid discs. Now, if we stack a whole bunch of thylakoids together, we get something called a grana. And a grana is just a way to organize and place a whole bunch of thylakoids into this internal chloroplast space. And then floating around, this diagram doesn't exactly show it quite like the mitochondria one did. Something very similar to the matrix that we saw just a second ago. This is called the stroma. It is this fluid-filled substance that sort of surrounds all of these stacks of thylakoid discs. So what is happening here? Chloroplasts are used to create C6H12O6, which we have seen time and time again, glucose, in the process of photosynthesis. Moving into our next set of structures, I would hesitate to call these organelles, but they are really critical structures. We're going to look at microtubules, microfilaments, and intermediate filaments. So microtubules, hear the word tube in this. What are tubes? They are hollow structures. And so microtubules are kind of like straws. They're going to have a hollow center. And so we are going to arrange these tubulin proteins in a pattern that allows for a hollow structure to exist. So this is a hollow rod or tube made up of even smaller tubes. And the protein here that is really important to associate with microtubules is the protein called tubulin or dynin. Those two proteins will create this overall tubed structure. Now, there are some critical things in the cellular world that are made of microtubules. And so there's something called cilia and flagella. If you have ever seen, let me see if I can pop up some pictures. Here we go. If you've ever seen a bacteria or even a sperm cell that is moving, you see this big, long, what looks like a tail. So this is flagella, a big, long tail that moves in a propeller-like fashion. That is a flagella microtubule. Cilia are typically a little bit smaller, and instead of moving like a boat propeller, they actually move up and down. They undulate, or they move like a wave in a wave-like pattern. Imagine taking your fingers, if you hold your fingers out in front of you and you just kind of wiggle your fingers, that's how cilia moves. Cilia beat, flagella rotate. And so we find cilia and flagella on lots of different kinds of cells. We'll see a few of those towards the end of this notes packet. But what's important here is what is in the green. So if we are trying to create cilia or flagella, the microtubules, the actual components of the microtubules, the tubulin, the dynin, they are arranged in what is called a 9-2 pattern, which means there are 2 of these tubulin proteins stacked on top of each other. And once we create a pair, we then put 9 of those pairs into a circular configuration to create the tube. Now, the other kind of tubulin arrangement is 9-3, which takes 3 of the individual tubulin or dynin proteins and sort of bonds those together and then arranges those takes 9 of them total, arranges those into a circular pattern that creates the tube. Now, we only find this 9-3 arrangement in something called centrioles in animal cells, and they are for the process of cell division. So keep in mind, microtubules, I'm recapping here, microtubules that will move are there for movement. We find examples in the cellular world of microtubules in cilia, in flagella, and also in these things called centrioles, which we'll see more about when we talk mitosis and meiosis. There is, this is a great picture here. So this is a centriole. These are two centrioles. Do you see how it looks like there's three stacks of protein, and then there are 9 of those stacks of 3 creating the overall tube? That's what the 9-3 arrangement really means. The next structure here is a microfilament. Now, microfilaments are smaller and they are not tubes, so they are not hollow. So these are solid rods of protein, and they are made of proteins called actin and myosin. And we see these in lots of different places, but specifically in the cellular world of muscle cells. So the idea that your muscles contract, that is the product of microfilaments, actin and myosin, moving against each other to create an overall contraction process. So this right here is the actin microfilament, and you can see it looks like a double chain that's sort of twisted. Each one of these little beads is called an actin. It's a protein. It's an actin subunit, and then you link them all together to create the overall actin microfilament. What is happening here? What is the function? This is typically a contraction movement function specific to pulling. And so if you think, well, pulling and contractions, that doesn't really make sense. When your muscles contract, they pull on your bones, and that allows your body to move. Now, these are a little cartoony representation of the actin that we see right here, and then the myosin, the other microfilament protein. This is what the inside of skeletal muscle cells look like. And so the contraction process happens when these myosin jump up, make contact with the actin, and push on the actin, and the whole muscle eventually will contract. This is not a muscle. This is something totally different. This is an amoeba who has used microfilaments within its boundaries to extend what are called like fake feet to enclose this little food particle. And so these pseudopodia, which is the fake feet, the pseudopods will extend out these microfilaments, will pull food back into their body, but they'll also extend out microfilaments and then pull themselves in that direction. So the organism gets overall movement by pulling these microfilament feet, and they move in the direction of those feet. So the last kind of filament, we've done microtubules, microfilaments. Here are intermediate filaments. So they're called intermediate because they're in size perspective, they're kind of in between the big tubes and then the smaller microfilaments. What are they going to do? I know that we typically talk structure. For that conversation, I want you to see all these little stringy things. This is a cell. This is the nucleus of the cell. And then you got all these little stringy things and also these strings kind of connecting outside of the cell. These are intermediate filaments. And so one thing that intermediate filaments are going to do is maintain the overall shape of the cell. Intermediate filaments will also fix certain organelles into position with each other or in position within the cell. So the nucleus isn't roaming all over the place. It's sort of anchored in position right here and held in position by these intermediate filaments. They also, what's really cool is these intermediate filaments provide almost like a series of jungle gym obstacle course opportunities for things to move around in the cell. And so various components within the cell can actually use these intermediate filaments and kind of walk to different locations within the cell by holding on to these filaments and using them as a guide. These intermediate filaments are the primary component of something called the cytosol. skeleton. This literally translates as the skeleton for the cell. And so you think about your own skeleton that provides structure, provides movement, provides support. The intermediate filaments are creating the skeleton of the cell, providing the exact same things. All right, we will pause here, and this will pick up in the last video, video number 3 for this notes packet.